This is Christina Fiore of MedPage Today reporting from the liver meeting in Boston. Researchers here have pared down animal livers to their vascular structure and then repopulated them with human liver and endothelial cells. This could be a small step towards the creation of bioartificial livers, which could help alleviate the current organ donor shortage. Dr. Pedro Baptista of Wake Forest University presented the findings here.
You have 110,000 people waiting for organs, being that kidney, livers, etc. From those, 16,000 are waiting for a liver. And if you count that 10 to 20% of those people never see an organ and die while waiting for one, then we certainly have to address that issue by trying new strategies. And one of them is certainly organ bioengineering that we hopefully will use for transplantation.
One means of organ bioengineering is washing away the cellular components of organs, leaving behind a vasculature and an extracellular matrix.
You basically put a cannula in the portal vein or the vena cava, and then you perfuse a detergent solution, which in our case is Triton X100 with a little bit of ammonium hydroxide. And this solution is able to really completely wash away the cellular components and the cellular membranes and even the nucleic acids and cellular proteins away from the scaffold, leaving behind only the extracellular matrix. The interesting thing, though, is that the extracellular matrix keeps its microarchitecture and its shape. So by doing this, then you really obtain something that at the end of the process keeps the matrix chemistry and has the shape of the organ that we intend to bioengineer.
The technique has been used before, but hasn't involved reperfusion with human cells. Baptista and colleagues seeded the scaffold with human liver progenitor cells and endothelial cells.
And then it's amazing because the cells recognize the matrix or the chemistry of the matrix on their own and localize and attach in what we think are their native niches, which means the endothelial cells attach into the vascular structures and the hepatocytic cells, if you want, attaching into more parenchymal areas. So they really recognize what's on the scaffold, which is a good indication that the whole processing to generate these cellular organ scaffolds, it still keeps most of what the extracellular matrix of a native organ is.
To test cell function, the researchers measured specific biomarkers, including albumin, urea, von Willebrand factor, and nitric oxide.
When we compare that with the cell function we have in cells seeded in a 2D environment, like a petri dish, we clearly see higher function, both from the endothelial cells we seeded and the hepatocytes or hepatoblast cells that we seeded. We clearly see higher function, which shows that once they start to communicate in this 3D environment, they definitely gain function.
Yet the challenge is getting enough cells to take hold. The Wake Forest team filled only about 30% of their scaffolding.
One of the major challenges, though, is that if the endothelial cell seeding is not efficient or not efficient enough, we will have blood clotting. And that's something we need to address also by the use of other techniques or other strategies in order to keep the blood flowing into these bioengineered organs.
Baptista said they plan on addressing that challenge by upping the number of reperfused cells from about 100 million to 300 or 400 million. At the liver meeting in Boston, I'm Christina Fiore, MedPage Today.